Of this presentation is on Lynch syndrome. Lynch syndrome is a genetic condition that increases the likelihood of an individual developing cancer. So let's talk about what this disease is. Begin by first looking at what cancer is. Cancer describes the uncontrolled growth of cells in a given tissue in the body. Cancer starts with when key mutations are made to genes that regulate the cell cycle. Under normal conditions, proto-oncogenes, shown right here, signal cells to divide when it is necessary for them to divide. Some mutations will mutate these proto-oncogenes into oncogenes. When a proto-oncogene becomes an oncogene, the signal to initiate cell growth and cell division, that signal is continually on. It's always on. It's always sending that signal for it to divide. And tumor suppressor genes, under normal condition, these genes, or the products of these genes, they act as certain checkpoints or blockades in the cell cycle. If certain conditions aren't met, such as insufficient DNA replication, cell replication stops. However, when these tumor suppressor genes are knocked out, that safety mechanism is no longer available. Down here, this is a figure showing the growth of cancer cells. If you notice, they grow uncontrollably. The size is very ununiform compared to these normal cells. The right here shows a hypothetical situation of the accumulation of mutations to take a healthy somatic or normal body cell to change it into a cancer cell. For instance, this one had a mutation knocking out one of the negative cell cycle regulator or a tumor suppressor gene, and then each successive mutation until it eventually becomes a cancer cell. Slide showing colorectal cancer. So again, we get the same thing where we have uncontrolled growth of cell. In this case, it's in the serosa of the colon epithelium. Here, this dye shows cancer development. And down here, we can see as tumors get more cancerous as they grow, they have a greater risk of what we call metastasis, is when these cells begin to migrate away from the initial tissue of origin and start moving to other parts of the body. That is when cancer becomes very dangerous, and we want to stop cancer before it gets to that point. Basic understanding of what cancer is, let's talk about Lynch syndrome. Lynch syndrome is a hereditary condition in which the risk of developing cancer, specifically colorectal and endometrial cancer, is extremely increased. It is inherited in an autosomal dominant manner. This means that you only need one copy of the diseased allele to inherit the disease. So 50% of offspring of an affected individual are expected to inherit this condition. It is caused by deleterious mutations to any of the genes involved in DNA mismatch repair. We'll talk about that more on the next slide, but this DNA mismatch repair is the spell check of DNA replication, and these genes are also classified as tumor suppressor genes, as we talked about in the previous slide. Lynch syndrome accounts for around 2 to 4% of all colorectal cancers, but individuals with Lynch syndrome can have up to an 80% risk of developing cancer in their lifetime, specifically colorectal cancer. So it's not a guarantee that you will develop cancer, but it is much higher risk. It shows us the mismatch repair system. The mismatch repair system can be thought of as the cell's spell check for DNA replication. Just by random mutation, as DNA polymerase goes on and replicates DNA, there will be mismatch bases. So there are individual protein complexes encoded by MLH1, MSH2, PMS2, and MSH6 genes. These genes encode for protein complexes that form a protein complex that comes together and repairs mismatch bases. If any one of these genes has mutations that makes one of these subunits no longer functional, these subunits are unable to come together to get rid of that mismatch base. So as a result, you get the accumulation of more mutations because you do not have this spell check, sort of say, acting on fixing any of these mismatch bases that occur from DNA replication. Consequences of mismatch bases not being repaired is you get something called strand slippage. So strand slippage, as you can see from this figure, in this case, it kind of bubbles up. And there's two scenarios here. You have a mismatch base here. You can either get a insertion where you pull in extra nucleotides because the extra strand, it'll fold up on itself and go that way, or it will do the opposite where the other strand will bubble up. And so as a result, you get these insertions or deletions in the DNA, and this happens at a much higher rate in cancers that have this mismatch repair deficiency as compared to normal cancers or cancers that don't have this mismatch repair deficiency. So the strand slippage that comes from these mismatched bases, you will get eventually as the cells keep on dividing uncontrollably, you'll have large insertions and deletions. This figure shows a little bit about what's happening. Here you have a large portion looping onto itself, and so it's during elongation you have more insertions. Then down here you can have looping going on in this direction. So this right here, this shows what is called a microsatellite repeat region. It's also known as short tandem repeats, if you've heard of that before. What it is, we have areas in our DNA that are repetitive, and there are a certain amount of repeats. And so in a given cell, we should have the same number of these repeats as every other cell in our body. However, a cancer with we call it microsatellite instability, the number of microsatellite repeats is different than in the tumor as it is from the cells in the rest of the body. This is indicative of a mismatch repair deficiency, which is a deficiency in that mismatch repair system, and it also makes the genome much less stable. This increases the rates of mutation, and it also increases cancerous effects because now the chromosomes are going to be much more abnormal, and they're going to, as a result of these different amounts of repeats, detect if an individual has mutations to any of the MMR genes. And so an individual with Lynch syndrome, which is deleterious mutations to any of these MMR genes, they can regularly get screened for colon cancer through a colonoscopy procedure. It is recommended that a Lynch syndrome patient receives a colonoscopy once every 2 years after they turn 18 years old. So regular screening can catch early cancer development before it even becomes a problem, before it becomes malignant. And this significantly increases the chances of survival and also a fully healthy recovery. Briefly about the current research and advancements that have occurred in the last few years, there is a vaccine for Lynch syndrome that is currently in development. So mismatch repair deficient cells, they actually generate high amounts of a tumor-specific neoantigen known as neoAGs. So this vaccine, what it aims to do is it delivers these neoAGs to the patient through injection, and it trains the T cells in our body to target cancer cells that express these neoantigens, so that once these cancer cells start developing, the T cells are able to take care of them much more early on in the process. Very fascinating disease, and it's also very important that we understand what it is because so many lives have been saved by early diagnosis and detection. My family, in particular, have been diagnosed with this disease. I have this disease, and so many members of my family have their lives have been spared because they have been tested and they've caught cancer early on before it has become a problem. If you'd like to learn more information, I'd strongly encourage you to visit the CDC's website on Lynch syndrome. Here is a link. Reach out to them with more questions, and I hope that this presentation has given you a good understanding of what Lynch syndrome is and how we can go about improving the lives of those with this condition.